Dr. SuJuLin, and is here? Oh, she's here. Sorry. Okay, you're going to show about understanding the mechanism of calorie restriction.
Okay, I first like to thank the meeting organizer for giving me the opportunity to talk here about our work. So our work is actually focused on understanding the mechanism of calorie restriction using yeast as a model system. So a lot of things I said probably apply mostly to yeast cells at a cellular level. Okay, and so as you know that calorie restriction has been known to shown to extend lifespan in a wide variety of species, and also it's been shown to delay the onset or decrease the incidence of many age-associated diseases like cancers and diabetes. Okay. And many studies have been suggesting possible mechanisms that could mediate lifespan extension and other beneficial effects. And, So among these many factors, I just summarized a few. So many others are not shown here, just a very brief model, which include that increased stress response or homeostasis, mitochondrial activity or mitohomeostasis, and increased repair capability, including genome and protein repair, and also increased SuJuLin activity or increased metabolic fitness. All these have been suggested to contribute to calorie restriction, mediate lifespan extension, and other beneficial effects. So we use yeast as a model system to understand how lifespan could be regulated at cellular level, and just because it has a very well-established molecular genetic technique, and also has relatively very short lifespan. So it's much easier to study cellular lifespans. And also many longevity genes that identify in yeast that actually conserve in high eukaryotes. And for example, so this slide is showing that several of the many CR-related longevity factor and pathway that actually conserve, that have been identified so far, and that conserve between yeast, is shown on the left, and high eukaryotes, is shown on the right. Okay, so these factors including proteins, like those are shown in oval shape. Okay, in addition to this protein, including the TOR pathway, and also the insulin sensing, glucose sensing pathway, and these are the protein-mediated sensing. And in addition to this protein, and also other factors, okay, secondary factors, okay, they are factor regulating pathway, including mitochondrial activity, or stress response, or NAD metabolism. All these contribute to the CR-mediated lifespan of beneficial effects. Okay. And actually, so today I'm going to focus on two parts. So one part is that we study factors that could affect mitochondrial activity, stress response, and actually NAD metabolism. Okay. So as I mentioned, we use yeast as a model system, and this slide is just going to show you how we study lifespan in yeast, how do we measure lifespan. So this is so the yeast that we use is a budding yeast, single-cellular organism, and the daughter cell is actually smaller than mother cell. So we could actually separate mother and daughter cell by their size under microscope. Okay. So CR actually could extend both replicative lifespan, which means the replication or division potential, and also chronological lifespan. So the one I'm going to show is how do we define and measure replicative lifespan. So here is shown is the mother cell. It's shown as N here. It's bigger than the daughter cell, okay? And then we just count under microscope how many daughter cells that bud out from the mother. Then we kind of could quantitate the number of cell division that each mother cell could undergo before they die, okay? And this is how we define replicative lifespan or division potential, okay? And the graph on the lower left shows a very typical lifespan curve, or replicative lifespan curve. And the CR condition that we use in the laboratory is by limiting the glucose concentration. And I want to stress that there are several CR regimens, and the CR model we focus in the lab is by reducing the glucose concentration. Okay. And as you can see that under CR condition, the replicative lifespan is significantly extended. And you could also see that a typical yeast strain, it could actually divide about 23 or 24 times. Okay. Now we could also measure another type of lifespan, which is to measure how long this cell could stay alive when they're not dividing. It's kind of similar to chronological lifespan or post-mitotic survival. And then what we do is just grow cell to stationary phase. Then we either shift them to water or leave them to culture for many days. And after several time points, it could take up to a couple of weeks. And we kind of take a sample at different time points and measure the ability for this cell culture to divide again. That's how we define. Means if the cells are viable, they're able to form colony again. That's how we define chronological lifespan. And again, under CR condition, the yeast cell also have a kind of extended post-mitotic or chronological lifespan. Okay, so many mechanisms have been suggested for CR, and the part that we focused on actually center on mitochondrial activity and NAD metabolism. And we previously shown, and also by others, that mitochondrial activity and the factors that regulate NAD-NADH ratio, which are also important for regular mitochondrial activity, play important role in CR. And activation of this factor actually lead to the increased activity of longevity factor, including SuJuLin, and some other metabolic longevity factor, which require NAD as a cofactor or co-substrate in their biochemical reaction. Okay, and these factors oops. Okay, thank you. Okay, all right. Okay, so I probably need 2 more minutes. Okay, so this factor probably extend lifespan by increasing stress response stability or genome protein stability or increasing metabolic fitness, and therefore lead to lifespan extension. Okay, and because okay, so we focused on because NAD metabolism obviously will contribute to the regulation of mitochondrial activity and this NAD-NADH ratio. So we also focus on NAD starting factors that would affect NAD metabolism, and also we like to identify a factor that could increase mitochondrial activity. And we first focus on the mitochondrial activity factors. Okay. So in order to identify mutants, so we mostly initiate our study from using genetic methods. Okay, so in order to identify mutants that would show, means when they have a gene that mutated that would show increased mitochondrial activity, we need a marker that allow us to easily screen these mutants. Okay, and so the first thing that came to our mind was maybe we could use nitric oxide, or the level of nitric oxide as a marker, because it's been previously shown that in mice, CR actually promote mitochondrial biogenesis in specific tissues like brain, liver, heart, and brown adipose tissue by inducing eNOS expression. Okay. And therefore, it was suggested in this study that CR could induce its beneficial effect by increasing eNOS expression and lead to nitric oxide production, and which will mediate mitochondrial biogenesis and SIRT1 expression. Okay. So then we want to know that whether this is also true in yeast by testing whether nitric oxide level is increased under CR conditions. And if so, we could use this a marker to screen for mutant that show similar phenotype. Okay. So this is the question we asked first. Does CR indeed increase nitric oxide level in yeast cells? And we use a fluorescent dye, as shown at the bottom. Oops, here. Okay. And this fluorescent dye has been shown actually will react with both nitric oxide and its oxidation product, including nitrite and nitrate. And it's been shown to be relatively specific to these molecules. And using this specific dye, we found that yeast cells grown under CR condition, as shown on the white bar here, actually show about twofold increase in relative nitric oxide level. And also this increase is transient. We only see that at the early growth stage. After 60 hours, then we do not see the increase anymore. So it suggests that E cell under CR shows a transient increase in nitric oxide level. So it suggests it could function as some kind of signal. And we also measure whether this is true in other mutants that have been suggested to mimic CR or like CR genetic mimic, including the TOR1 mutant. Oops, keep pressing the wrong button. Okay, right here. And also HSK2 mutant is a mutant defect in glucose hexokinase. Okay, and so both actually behave similar to CR cells. They show kind of about a moderate increase in nitric oxide level. Okay, so the conclusion for this slide is the nitric oxide related signal are actually increase in E cells grown under CR. Okay. And this increase in nitric oxide is not due to the fact that the cells are unable to detoxify nitric oxide, right? So there's two possibilities, right? So cells have higher nitric oxide, then maybe they are kind of compromised in the detoxification mechanism. But actually, we found that cells grow under CR, is shown here, and also the CR genetic mimic, the TOR1 and HSK2 mutant, they're actually more resistant to nitric oxide treatments. So the experiment shown on the left is we treated the cell with the S-nitrosyl glutathione, is a nitric oxide donor, and we found that all these cells actually are more resistant. This is the growth at different concentration of GSNO added. They are more resistant relative to wild-type cells. And then we also measure the enzymatic activity of two major nitric oxide detoxification enzymes in yeast, which are SFA1 and YHP1. And we found that actually these cells, E cells actually have slightly increased YHP1 activity. So the data from both experiments suggest that these cells under CR, they have increased nitric oxide level, but they actually also have enhanced nitric oxide detoxification activity. Okay, so then now we have a possible marker. Okay, so we want to use this fluorescent dye to identify yeast mutant that possibly show increased nitric oxide level, similar to CR, and then we're hoping that maybe some of this mutant could be another group of CR genetic mimic, or mutant that would show enhanced mitochondrial activity. So then we took yeast genetic gene deletion collection. Then we screened for mutant that have higher nitric oxide, and we came up with about 150 mutant with high nitric oxide. Then we screened for those with another criteria is they cannot have growth defect, and also they should also show resistance to nitric oxide donor. So then this way they're kind of similar to CR mimic. And also we are less likely to get those mutants that just have defect in nitric oxide detoxification, because we do not want to get this group of mutants. Okay, so then after that we come down with 17 mutants that kind of pass this. Then we kind of sort this 17 with their high nitric oxide level. Then we did chronological lifespan and replical lifespan, and we found that about 14 of them have either extended chronological lifespan or replical lifespan, and only 5 of them with both extended chronological lifespan and replical lifespan. So more like CR conditions. Okay. Right. So these are the 5 mutants, and I will not focus on the detail because we still don't know how they work yet. So we're gonna focus on understanding how this mutant would would they really function in the CR pathway and how they regulate lifespan. But I'll show you a few slides to summarize the phenotype of these mutants. Okay, so they all have extended replical lifespan, chronological lifespan, and most importantly, because we try to find mutants that also have high mitochondrial activity, and indeed, when we measure their oxygen consumption activity, which is a marker for mitochondrial respiration activity, and all these 5 mutants also show increased oxygen consumption. Okay, so this mutant, without knowing the detail how they work to extend lifespan, we cannot say they are true CR mimic, but at least we could kind of have a preliminary conclusion, which is there's a potential correlation between the between increased nitric oxide and mitochondrial activity and CR-induced longevity at cellular level. Okay, so now you're probably thinking that, where does nitric oxide come from in yeast, right? Because there's no eNOS in yeast. And although there's been reported eNOS-like activity in yeast, but the gene responsible for this activity has not been identified yet. Okay. But since it has been shown that actually the mitochondria respiratory component, the COX, complex 4 here, okay, which is short for COX, stands for the complex 4. Actually, it's been shown to produce nitric oxide, but this activity is more obvious under hypoxic condition. Okay, so maybe this is a possible candidate, but we did not grow cell under hypoxic condition, so we did not know whether it's true in our model. Okay, so you know that this complex 4 normally will just deliver the electron to oxygen and produce water, and this is normally how they work in electron transport under normal condition. But this same complex could also produce nitric oxide using nitrite as a substrate, and lead to nitric oxide production. Okay. And this phenomenon is not just specific to yeast. It's been shown that in mammalian, mitochondria purified from some mammalian cells also produce nitric oxide or increase production of nitric oxide under hypoxia. And this activity seems to be NOS independent because it cannot be inhibited by a typical NOS inhibitor. Okay. Oops. Okay. So then we ask if COX is really responsible for nitric oxide production in our system. Then eliminating the activity of electron transport should also abolish this nitric oxide production in CR cell. And as shown here, this is a wild type cell under CR. You see the increase of nitric oxide production. And we delete CYT1, which is the cytochrome C1, okay, of the electron transport component. If you delete this gene, you block the electron transport. And you can see here, oops, right. So after deleting this component, the nitric oxide production is largely eliminated. And I want to point out here, there's still some increase of this mutant on the CR. So that would suggest that there's some other source of CR production on this, of NO production on the CR. But it seems that mitochondrial respiration will really contribute to nitric oxide production in yeast. And the slide on the right just shown that this activity is not, I would say it's not NOS dependent, because this activity is shown here. The increase of the nitric oxide production is not inhibited by a NOS inhibitor. Okay. All right. Okay. Then we ask, well, if this activity is enhanced under hypoxia, right? And would that does yeast could actually produce nitric